Toronto General Hospital Health Research Institute. I'm studying the gut-brain nutrient sensing on feeding regulation. Around 2 years ago, I did my PhD in nutritional science here, U of T, also studying something related to micronutrient like vitamins. Hopefully, after today's lecture, you all learn something you didn't know before. So today's topic will be vitamins. As you can see, we categorize the vitamins into water-soluble and fat-soluble vitamins based on their solubility. So this week's module, there's 2 sections of those. And before we started, there are 2 supporting documents associated with this module 6 and 7. Supporting document 6, which include a brief introduction of the water-soluble vitamins. Specifically, there's vitamin B1, 2, 3, thiamine, riboflavin, and niacin. Those 3 vitamins are only discussed briefly because they are found more than adequate in Canadian diet, so it's not really a concern about their intake. And also the supporting document 7, there'll be a discussion on the interaction between vitamin C and E, specifically how they interact to protect us from oxidative stress. One important theme in our discussion of vitamin and also later discussion on minerals is that those micronutrients play their role in a combination. So the supporting document also emphasizes how several B vitamins, including thiamine, riboflavin, niacin, pantothenic acid, also called vitamin B5 sometimes, and biotin, how they function in the energy metabolism. And also, we'll learn about how vitamin C and E they both are antioxidant in our body, how they protect us from oxidative stress and oxidative damage. And last but not least, we'll look at the role of 3 important B vitamins, including vitamin B6, B9, also the main name of B9 is a folate, and also B12. How they play a role in the one-carbon metabolism or single-carbon metabolism. The why we call it one-carbon metabolism because this includes a series of reaction and is involved in transfer of the methyl group from one unit to another substance, which we will touch about in details later. So before I introduce the vitamins, I just want to first show in this digestion and absorption figure again, just to introduce how those vitamins are absorbed in our body. So basically, when after we eating our food, normally contain macronutrient and micronutrient. So in micronutrient, it includes vitamins and minerals. So today, of course, the topic will be in vitamins. So in vitamins, water-soluble vitamin and fat-soluble vitamin. So the water-soluble vitamin, they are just easily diffused into the gut. So the main site of the absorption of the water-soluble vitamin is the duodenum, which is the upper part of our small intestine, and the jejunum, which is the middle section of the small intestine. But for the fat-soluble vitamin, the process of its absorption is a bit more complicated because its absorption requires fat. So if you still remember how the fat absorbed in the previous sections, so it mainly is incorporated into my cell, which showed here is a complex consisting with fatty acid, bile acid, and fat-soluble vitamins. And then this structure my cell diffused into our gut cell, which was also called enterocyte, and where chylomicron can be formed. So if you still remember, chylomicron will be transported into the lymphatic system rather than blood cell to be utilized later. So in general, water-soluble vitamin goes to the blood, and fat-soluble vitamin goes to the lymphatic system for further utilization. So let's start with the water-soluble vitamin. So in this section, we're going to be mainly focused on the vitamin B6, folate, the vitamin B12. So let's start with vitamin B6. There's three main reactions in which vitamin B6 play a role. This figure here shows that the vitamin B6 play an important role in metabolism of amino acid. If you still remember before when we discussing the amino acid part, amino acid consists of amino group, shown in blue here, and a carboxyl group, shown in purple here. And vitamin B6 is a coenzyme for the enzyme that catalyzes those reactions in the amino acid metabolism. So what is a coenzyme? So basically, a coenzyme is an important structure for the enzyme to be able to function. A good analogy would be if you consider an enzyme as a car, the coenzyme will be the driver. So you cannot move the car without a driver. That's how important the coenzyme is. And vitamin B6 is a driver of this, those series of enzymes. So in the first reaction, we showed here, the amino group, shown in blue, can be transferred from one amino acid to another. And this reaction is called transamination, which is required vitamin B6 as a coenzyme. And after being removed its amino group, the remaining carbon skeleton can either be converted to glucose through gluconeogenesis in the liver, or enter the citric acid cycle to be converted to carbon dioxide with generation of ATP. And this process is called deamination. And finally, the carboxyl group shown in the purple can be removed and leave the compound that ultimately will be converted to a neurotransmitter, which help to send signal between nerve cells. So this process is called decarboxylation, which also requires vitamin B6 as a coenzyme. To introduce the next function of vitamin B6, first we need to introduce the amino acid called homocysteine. So homocysteine is an non-essential amino acid that's believed to be toxic to our blood vessels. People have higher level of homocysteine in the blood have been shown to have an increased risk of cardiovascular disease. Vitamin B6, along with B12 and folate, are three vitamins that work together to keep our homocysteine level in check. This concept is part of what is called homocysteine hypothesis, which saying that increased intake of vitamin B6, folate, and B12 should reduce the risk of cardiovascular disease. And all three vitamin B vitamins are required to maintain low homocysteine level. And we'll see how this happens in next slides. So in this graph, we can see that three B vitamins function together to lower homocysteine level in the blood. So in the first pathway, which at the right of the slides, we can see that vitamin B6 functions to convert homocysteine, shown in orange, to cysteine, which is another amino acid. So it lowers blood. So this process itself lowers blood homocysteine level. And vitamin B6, again, play a coenzyme role in this process. And another reaction showed in process 2, vitamin B12, we have vitamin B12 and methylfolate. So methylfolate is a folate attached to a methyl group. And this reaction happens when the methyl group transfer to the B12, so which form the methyl B12. And then the methyl B12 will give the methyl group to the homocysteine to be able to form methionine. So that's another way to lower the homocysteine by transferring the methyl group. And finally, in the pathway 4, we can see that vitamin B6 have another role acting as a coenzyme in the reaction of folate forming the methylfolate. So in this, vitamin B6 catalyzing the conversion, and so that's the whole reaction shown in this graph can be sustainable and can keep going on. So this transfer of methyl group is what is called one carbon metabolism or single carbon metabolism. So as we just saying, the three vitamins are part of the homocysteine hypothesis. So how strong is the evidence to support this hypothesis? Well, of the three, folate is one of the most difficult to get from the diet. When you look at the literature, most of the study has looked at the folate and its effect on cardiovascular disease through it's understood. Although it's understood that folate is typically combined with vitamin B6 and B12. So what is the evidence? Based on the observational cohort study, we know that when folate in the blood increase, the cardiovascular disease decrease. But our observational study measures only association but not causation. The evidence in the randomized control trial showing that when folate supplement are taken, the level of homocysteine in the blood also declines. So what we are also known from cohort study is that decreasing serum homocysteine is associated with a reduced risk of cardiovascular disease. What is missing from the evidence is a confirmation from RCT trials that lowering serum homocysteine levels reduce cardiovascular disease directly. The last causal piece of the puzzle is still missing, so this hypothesis is still remain fully confirmed. It's still yet to be fully confirmed. We next move to folate. Folate is also called vitamin B9 sometimes, and there's two forms of folate. So the first one is folic acid. It's more from the supplement or it's a synthetic form, and it's mainly occurs in the supplementation and food fortification. It has a ring structure attached to a glutamate, which is an amino acid. And also we have a naturally food-formed folate polyglutamate, which has a ring structure attached to 5-glutamate. Of note that the synthetic form actually, when taken as a supplement form, it has 100% bioavailability, so it's really easy to absorb. But when it comes to in the fortified form in the food, the bioavailability reduces a little bit, around 85%. And the form in the food actually has much lower bioavailability than the synthetic form, folic acid. Folate, as we already seen in the earlier discussion on the homocysteine hypothesis, is involved in one-carbon metabolism. There's two main functions of the one-carbon metabolism that we will discuss here. One is the synthesis of component of DNA. The another involves addition of the methyl group to the DNA in a way that regulates gene expression. So in this figure, we can see that the variation of DNA methylation will actually alter the number of type of genes that are expressed and further affect protein translation. So DNA methylation regulates protein synthesis and cellular metabolism. Alteration of DNA methylation actually also referred as a type of epigenetic effect on our gene expression. So normally, at a sequence of DNA, adding a methyl group normally causes silence of the genes, but sometimes it also acts as a switch to turn on the gene. In this figure, mainly in the lower part, we can see that the role of folate played in DNA synthesis and how this role impacts the development of our red blood cells. When a folate intake is adequate, the DNA is being properly synthesized in the precursor red blood cells, and these precursor blood cells develop into normal blood cells. That's a normal physiology. But if a folate is deficient and the DNA synthesis is impaired, cells are not able to divide normally, and then they form large metalloblastic cells. Those cells sometimes contain nucleus, sometimes they don't have it. When they don't have it, they are referred as macrocytes. So folate deficiency resulting in development of anemia. So that's often referred as a megaloblastic anemia or macrocyte anemia. Folate also has an important role in the fetus development. It is needed for development of neural tube. The neural tube is a fetus, fetus eventually develop into the brains and the spine. And the figure on the right here shows that at day 19 to day 28 represent the day after conception. A neural tube defect normally results in development of serious birth defect, and that's going to affect brain and spine. And development of the neural tube requires folate, and low dietary folate intake, often in combination with a genetic predisposition, has been shown to increase those defects. Now, one of the problems with development of neural tube defect is that it often occurs before women typically know they're pregnant. So in order to prevent this happens, in Canada, after 1998, there's a mandatory mandatory folic acid food fortification, specifically in the stable food section. And a lot of stable food, including white flours, cornmeal, and pasta, are fortified by folic acid. And in addition, Health Canada also recommends that women who might get pregnant should take multivitamins that contain at least 400 micrograms of folate to prevent this birth defect. This graph illustrates that how folate functions in neural tube development. So it functions through an intermediate of amino acid methionine. Folate, vitamin B6, and also vitamin B12 are needed to generate methionine derivative. So this derivative then participates in DNA methylation through a one-carbon metabolism, although it's not showed in this graph. But this also needs all three B vitamins to transfer the methyl group to the DNA. And a proper DNA methylation resulting in development of a healthy neural tube. And the most common neural tube defect is spina bifida, which results in neural tissue being squeezed outside the spine due to the incomplete closure of the neural tube. This defect results in paralysis. A neural tube defect can affect the development of the brain, can also be fatal or cause serious irreversible impairment. And since folate fortification began, there has been at least 50% reduction in neural tube defect in Canada. So we can confidently say that this program has been very successful. But despite of the successful story, some people has also expressed their concern on if there may be too much folate in our food supply, or more specifically, folic acid. In this graphic, show the amount of folate this graph showed the amount of folate found in blood of Canadian women of a reproductive age. But what is, and this graph is called a cumulative percentage graph, and it's measured amount of folate in red blood cell across horizontal axis. And what we can see here in position 1 is that it's considered folate deficiency. So we can see literally there's no one in Canada has folate deficiency, or at least no women in reproductive age have folate deficiency. The second cutoff here is believed to be high enough to prevent neural tube defect. Is about, so you can see around 22% women has level lower than this. That means around 80% of women are consuming adequate amount of folate to prevent neural tube defect. And the third cutoff here is what is called high concentration cutoff. And we can see that at least 40%, here is 6%, but at least 40% of Canadian women has level above this high concentration mark. So we can ask the question, is folate intake in Canada too little or too much? I think we can conclude from this figure that we definitely not having too little. But also the U-shaped curve showed here to remind you that it's possible we're consuming too much. The U-shaped curve, dose response curve is characteristic of essential nutrients, for which either too little or too much of the diet can lead to adverse health effect. Let's continue with this question of too little or too much. of folate. So this graph, taken from study published in 2007, which showed the year on the horizontal axis against number of cases of colorectal cancer per 100K individuals on the vertical axis. As you can see from the graph, cancer rate were steady declined until the year 1997 or 8. That's a time when the Canadian government start the folate fortification amendment. Now, this graph does not prove that folate fortification causes increased colorectal cancer rates because after 2000, it seems this rate seems reduced. So no one really know what is going on about this bump. But for human cohort study, we saw that the increased folate is associated with decreased colorectal cancer. And when we look at animal study, the situation is somewhat more complex. So when you give animal folate in the absence of the pre-existing cancer, the animal experienced decreased cancer risk. However, when you give the animal high dose of folate in animal with pre-existing cancer, and their cancer cell grows even faster. So this may explain the bump here. Because folate has, as you still remember, folate has a role in the DNA methylation and the gene expression. So when you have a fasting growing cancer cell, adding megadose of folate may help to growth. That may happen here when someone have pre-existing cancer, and when you add much folate in the during the food fortification program, their cancer rate seems manifested. And after 2000, it seems that the reduced rates of this after 2000 seems still unclear. But if you see clearly here, the case is really from 52 to 54, only 2 cases increase out of 10K. So it's not really suggesting the folate fortification really caused the cancer. And also, we already know it's a really successful program to prevent neurotube defect. But however, the data does suggest that we need to be cautious when about fortifying nutrient in our food supply. This is an example highlighting the need for public health and examining all the potential risk and benefit before you mandate the fortification. Some other example from preclinical data, specifically in rats. In the past decade, Dr. Harvey Anderson lab in nutritional science, where I also did my PhD from, conducted a series of study in rats showed that increased gestational vitamin supplementation, especially folic acid and other B vitamins, lead to increased risk of development of obesity and metabolic disease in offspring, raising the possibility that the overconsumption of folic acid, especially in the prenatal supplement, also maybe contribute to this current obesity epidemic. The last B vitamin that will be discussed here is vitamin B12. Vitamin B12 deficiency results in development of what is called a secondary folate deficiency, which produces the same megaloblastic anemia that occurs with folate deficiency. And we will be discussing this in a moment and see why this is the case. And vitamin B12 is an interesting vitamin because it can be produced by microorganisms. So these microorganisms are consumed by animals. Then the vitamin B12 will be accumulated in the animal tissue. Thus, animal products or animal food actually is a good source of vitamin B12. That's why when people consume a full vegan diet, they need to have vitamin B12 supplementation. The absorption of vitamin B12 is quite complex compared to other water-soluble vitamins. So vitamin B12 is normally with a protein in the food, and when is ingested, the stomach acid and the pepsin, which is an enzyme in the stomach, are required to break the bond between vitamin B12 and its bonded protein. In addition, the stomach also secretes a protein called intrinsic factor, and this intrinsic factor is required for vitamin B12 to be absorbed in the intestine. So after vitamin B12 is released from its bonding protein, it will go to the small intestine from the stomach along with the intrinsic factor, where they can bind together and be absorbed through binding its specific receptor in the small intestine. And in the colon, actually, our microbiota can also synthesize this vitamin B12, but it's still unclear whether this colorectal biosynthesis vitamin B12 can be used for us. And that's the absorption of vitamin B12. And let's move to the left side of the slides. The pernicious anemia is actually caused by inability of absorb sufficient amount of vitamin B12 due to an autoimmune condition that destroy the cell that produce the intrinsic factor in our stomach. So the pernicious anemia have the same symptom as the megaloblastic anemia because both vitamin B12 and folate are responsible for our red blood cell formation, and in the same way. But pernicious anemia specifically indicates an anemia due to an autoimmune condition. So not surprisingly that pernicious anemia won't respond to any iron supplementation because it really have nothing to do with iron status in our body. Let's look at the case study at the start of chapter 8. Is describing a case that in an elderly gentleman, Mr. Chen, who developed a neurological symptom. The neurological symptom was associated with he has tingling and loss of balance are due to the role that vitamin B12 play a formation of the myelin sheath, which is a neural structure to transmit the signal to the brain. And this gentleman, Mr. Chen, in this case study, actually had atrophic gastritis, which is a condition that caused by gastric inflammation, and it has reduced secretion of stomach acid. The reduced stomach acid, if you remember, the absorption of vitamin B12 is required. It requires the stomach acid to break the bond between the B12 and its bonding protein. If someone has reduced stomach acid secretion, that means this process cannot be fully finished. And also that will explain the neurological symptom observed here. So Mr. Chen probably have vitamin B12, insufficient amount of vitamin B12 absorbed due to this gastric condition. And the way to solve it is to treat this symptom with vitamin B12 intravenous infusion and following by the megadose vitamin B12 supplement. So actually the supplementing vitamin B12 is not bound to protein, so it will be easily absorbed. And also when using high dose vitamin B12 supplementation, even without an intrinsic factor, there is still sufficient to be passive absorbed to prevent this vitamin B12 deficiency. Now, this question is really about which treatment approach in people with obesity will require vitamin B12 supplementation to prevent deficiency. So as you know, historically speaking, the most effective way to treat obesity is actually the bariatric surgery. Maybe a lot of you already know the Ozempic drug, but before that, bariatric surgery is the main way to go. The procedure involved in cutting a part of the stomach of the patient. It's not only it's helped weight loss, not only because it's reduce size of our stomach, so you eat less. But also it involves a lot of complex mechanism, including different profile of your gut hormone will affect your appetite. In that case, if someone get a part of their stomach removed, they will affect their gastric stomach acid secretion. Also, the part removed also affect their intrinsic factor secretion. So in that case, it's no-brainer that the patient with bariatric surgery may need vitamin B12 supplementation. And the second case is the patient with GLP-1 based drug, which you might heard about Ozempic. The patient with Ozempic or other GLP-1 based medicine injection, they have reduced appetite. It's also a mechanism that's pretty similar with bariatric surgery. So this type of drug, they mimic our endogenous GLP-1, so it's a type of gut hormone, and travel to the brain to bind to the receptor to lower food intake. But this food lowering effect also associated with some changes in preference of some certain type of food. So patient with GLP-1 drug also may have all sort of sufficient nutrient intake. So not only vitamin B12, maybe other micronutrient supplementation should be considered. So too much folic acid has been found to masking the B12 deficiency. So what this means is that pernicious anemia does not develop, and this anemia is an early warning symptom of a vitamin B12 deficiency. But neurological damage continues to take place. The UL of the folic acid is set to 1,000 microgram, and also that is a dose that's low enough to prevent this masking of vitamin B12 deficiency. And we'll see why in the further slides. So let's take a look how this masking of the vitamin B12 take place if you have too much folic acid in your system. These heavy black bar indicate that reaction that would block the absence of vitamin B12. And you can see that without vitamin B12, the conversion of methylfolate to folate does not take place. And instead, there will be an accumulation of the methylfolate. And as we know, under normal intake of folate, vitamin B12 deficiency would impair DNA synthesis and red blood cell formation, resulting in anemia. And also, this is an early sign for vitamin B12 deficiency. However, vitamin B12 deficiency in the presence of a very high dose of folate intake will not result in anemia. That's because with very high dose of folate intake, the need of vitamin B12 is mediated. The need for vitamin B12 mediated conversion of the methylfolate to folate can be bypassed. So although there's no anemia sign, the neurological damage caused by vitamin B12 deficiency continue to take place. We can see here that absence of vitamin B12 prevents conversion of the methionine to a methionine derivative that functions in myelin sheath formation, and under vitamin B12 deficiency, neurological symptom can still develop. Here is some different forms of micronutrient deficiency, although we didn't cover all of those here. Just want to show you how those what was a symptom of certain micronutrient deficiency. So first here we have scurvy. Scurvy is a symptom of vitamin C deficiency. It's normally involved in bleeding gum, weak teeth, and in the middle we have xerothymia, which normally involved in the scarring tissue developed around the corner of the eyes. It's caused by vitamin A deficiency. We will discuss more in the later slides, in the later part of the vitamin A part. And we also have goiter here. It's a deficiency of iodine. Normally, the patient with this type of deficiency will develop a huge neck. That's because their thyroid gland is expanded. and for in the children, this is called cretinism. Is because of the is a result of the congenital hypothyroidism. And at the left part, we have cricket, which is a vitamin D deficiency. Normally, we call it cricket in children, and unfortunately, this symptom can be reversed by vitamin D supplementation, which we will also discuss more in the vitamin D part. And this concludes this section 1. To assist your learning, and please try look at the self-assessment question at the end of the module. And in the section 2, we will continue to discuss the fat-soluble vitamin A, E, D, and K. So we can start with vitamin A. Vitamin A is the most common deficient vitamin in the world. Vitamin A is present in a limited number of food, but both in animal food and plant-based food. And if the population does not consume those food, they are vulnerable to the deficiency. Here we see the upper part, the CCSH data on the vitamin A intake. The graph shows that the vitamin A intake for men and women indicate that the portion of Canadian adults intake of vitamin A is below EAR. And we can see that this is over threshold to the 10%, indicating that there are many Canadians who are not meeting the requirement of vitamin A. And this also a reflection of the fact that the most Canadians do not consume enough fruit and vegetables or dairy products, which are all good source of vitamin A's in a diet. And this graphic is a typical fast food meal, which does not contain very high amount of vitamin A, except there might be a piece of cheese, which is a dairy product containing some vitamin A. And of note that for dairy products, normally full-fat dairy contain a high amount of vitamin A, but Health Canada also mandate the reduced fat dairy products such as skim milk or like 2% milk. They are mandatory to have vitamin A fortification. So for vitamin A, there are actually two types of it. So first is preformed vitamin A. We also call it retinol. So this is the main bioactive form in our body. It's mainly from animal products, including dairy or yolk or egg, specifically yolk part. Also, the liver is a main source. If the liver is your part of a diet, it include retinol, ester, and retinol. And also for the plant-based, it's called provitamin A. It's called carotenoid first of all, and it include beta carotene, alpha carotene, and some of the carotenoid can be converted to the retinoid, which is the main bioactive function form in our body. And please note that not all carotenoid can be converted. Now let's look at the structure of the previtamin A retinol. So retinol consists of a whole bunch of molecules. They are pretty structure similar. For the basic structure, they all have a beta ionol ring here attached to a polysaturated side chain. And if you add a hydroxy group to the end of the side chain, we have retinol, which is a main transporter form in our body. And when forming the aldehyde, it's called retinal. It's an essential form for our vision, which we'll spend some time on that later. And by adding carboxylic group or ester group, we have retinoic acid and retinoic ester. So retinoic acid is mainly function as a gene regulator in our body, and the retinoic ester is a main storage form in our body, specifically in the liver and in our adipose tissue, which means fat tissue. As for the plant-based, plant-formed carotenoid, they are the precursor of the vitamin A, meaning they can be converted to the preformed vitamin A retinol, which is it introduced. The meaning, and the way they have bioactive function is mainly one part is after this conversion to the pre-vitamin A, but also it can act as antioxidant in our body. And so far, there are more than 600 of them out there, this carotenoid, but only about 50 of them have bioactive function of vitamin A. And structurally wise, they all have expanded carbon chain with conjugated double bonds, and typically with a beta ion ring at one or both sides of the chain. A few things need to be mentioned about this conversion of beta carotene to or carotenoid to the retinol. So as we use beta carotene to retinol as example. The conversion is not very efficient. First of all, it's not like all the beta carotene can be absorbed, and even absorbed, it's not all the beta carotene can be converted. So there's a lot of factors involved in this conversion, including because also this conversion needs an enzyme, also require other nutrients. So and also genetic is also a factor. Some people have polymorphism, genetic variation, which affect this conversion in different degree. Because of this conversion of efficiency of this conversion, in order to produce 1 microgram of retinol, you will need to consume 12 microgram of beta carotene. This is expressed in the unit called retinol activity equivalent, RAE, rate. A 1 microgram of retinol is a 1 microgram of RAE, and this is in turn can be derived from 12 microgram of the beta carotene. From the beta carotene, for beta carotene as are not converted, they typically circulating in the blood and act as antioxidant. And in the late 1980s and late 1990s, a number of cohort studies find that a high level of beta carotene in the blood were associated with a reduced risk of lung cancer in smoker. However, the positive story of the beta carotene is not the case in an interventional trial called alpha-tocopherol beta carotene cancer prevention study. So the alpha-tocopherol is a form of vitamin E, which we'll talk more in the vitamin E section. And this intervention trial looked at how vitamin E and its role in the cancer prevention. And they found there's no effect of vitamin E on cancer. And this study also gave a large dose of beta carotene to the smoker over the period of about 7 years. And the result of the study showed that beta carotene group compared to the group with no supplementation has actually increased risk of lung cancer by about 17%, which is quite substantial. The problem of this study might be that the dose of the beta carotene they use is pretty high. When someone is over-consuming the beta carotene, this beta carotene, the majority of them, first of all, won't be absorbed, won't be converted to retinol, preformed vitamin A. And the rest of them will actually accumulate it in the fat tissue underneath the skin, turning the skin color like orangish. In this trial, about 30% participants have this orange skin symptom. So fairly high dose were used in this trial. And also it might be important to add that it's very unlikely that you consume this amount of beta carotene just from food. It may be achievable through megadose supplementation. And in the animal study, where you can dissect the mechanism, scientists actually find when animal has extremely high dose of beta carotene, they had an increased risk of developing lung cancer. However, when the dose was more moderate and close to the dose that can have seen in the diet, the beta carotene was actually protective. So this is another classic example of the U-shaped curve when too much something good cause harm. Thus, it's need to be cautious that and also mindful when you read papers, especially associative paper, and also always gather comprehensive information across the board before you draw a conclusion. Here I'm showing is a website, has more information on vitamin A, and please maybe take more time to go through this content later. And here also some question for you after you read through the introductory paragraphs of vitamin A. Now, let's look at how vitamin A functions in our vision. So here's our eyeball. It's actually like a small structure squeezed out from our brain to detect light. When here's like any light sources here, the light signal go through the cornea of the eye and hit the back of the eye where the retina lies. And the retina actually consists of 3 layers of a structure, including nerve tissue, rod cell, and cone cell. And both rod cell and cone cell, they are photoreceptors neuron in the retina and can convert this light signal to the electric signal and move this signal to the brain to generate image. That's how we have vision. And specifically in the rod cell, there's a highly light-sensitive G protein couple receptor protein called rhodopsin, like short structure here. You can see it consists of 11-cis-retinol, which is a form of retinol, and also the structural protein opsin. So when the light signal hits this receptor, rhodopsin, it goes through the transformation involving cleaving into opsin and cis-retinol, and then this cis-retinol convert to trans-retinol, and those two reunited and back to rhodopsin. So it's a cycle. And when there's an inadequate amount of vitamin A in the body, the rhodopsin cannot be properly synthesized. Thus, the light detecting will be delayed or impaired. Thus, it's causing the night blind. So vitamin A also controls gene expression over 500 of genes in our body. And there's 2 forms of retinol, all-trans retinoic acid and 9-cis retinoic acid are the main gene regulator. So if you recall retinol or vitamin A, they're fat-soluble, so that means they are hydrophobic. In order to move around in the aqueous phase in our body, such as blood or cytosol in our cell, there has to bond some protein or carrier to be able to move. In the cell or in the cytosol, those 2 retinol bond to their carrier called intracellular lipid-bound protein. And in the blood, if you also want to know, is retinol normally bound to the protein called retinol binding protein. So there's 4 different isoforms to help vitamin A or retinol to freely move in our body. And after they bind to their carrier protein, they move into the nucleus, which showed in this circle, and they then bind to each other's receptor. So all trans retinoic acid bind to the alpha, beta, or gamma retinoic acid receptor (RAR), and the 9-cis retinoic acid bind to its receptor alpha, beta, gamma, or alpha, beta, or gamma retinoic X receptor RXR. And together they drive the retinoic acid responding element, also called RARE, to promote gene expression and further protein translation. That's how vitamin A regulates our gene expression. Vitamin A also controls the differentiation of the epithelial cells. Specifically, this vitamin controls expression of genes that cause differentiation of newly formed epithelial cells into healthy mucus-secreting cells. If a person is vitamin A deficient, then the epithelial cell will not differentiate properly, and the mucus-secreting cell will become dry cell to produce a protein called keratin. The epithelial tissue are in the eyes, very small and large intestine, and also in the lung and other organs. The mucus created from the cell form a barrier against outside bacteria and reduce the risk of infections. Thus, vitamin A deficiency also increases the risk of infections. Here we see the condition called xerophomia, where there's an impairment in differentiation of the cell in the cornea of our eyes, causing dry eyes. And the cornea of the eyes becoming dry, cloudy, and in the extreme case, will cause blindness. And also vitamin A deficiency is a major cause of blindness in low-income country. In addition to controlling cell differentiation of the epithelial tissues, vitamin A is also required for differentiation of many of the cells that control the immune system. So precursor cells differentiate into many types of the white blood cells that requires fighting the infections. In the absence of vitamin A, the immune system is compromised. It plays an important role in both innate and acquired immunity. So in the innate immunity, white blood cell that's circulated in our blood is crucial for this immune response. Here we can see that there's two types of white blood cells. So we have phagocyte and lymphocyte. And phagocyte actually the first responder to the infection. Against infection, they actually engulf the harmful pathogen they detect in the blood, and to destroy this pathogen and then present is a protein on the pathogen to the lymphocyte. And this presenting protein is also called antigen. So it's an identification of this specific pathogen. And the second set of the cell, lymphocyte, will pick up this antigen, and we'll discuss here is called acquired immunity. When the antigen are presented by the phagocyte, the lymphocyte will begin to synthesize antibody. And this antibody then go to circulation and bind to harmful organisms. So when the body have this specific pathogen first, and it will engulfed by the phagocyte first, and the antigen or the specific identification of this specific pathogen will be presented to the lymphocyte, and the lymphocyte will remember this to secrete antibodies specifically just for this type of pathogen. So that's time if body will encounter this type of pathogen again, the floating antibody in our body will attack to this pathogen directly instead of just go through the first round again. So it's really faster and efficient way to help us to protect the pathogen. So vitamin A deficiency impairs this development of acquired immunity. In overall, vitamin A is important for all the immunity, including both acquired and innate immunity. Vitamin A supplementation prevents infection and blindness, as we mentioned before. And in this picture shown here, a healthy walker is squeezing a capsule of oil, which contains vitamin A, into the mouth of this kid. And the supplementation can provide 6 months of supply of this vitamin. So the supplementation is required only twice a year. The Nutrition International, also formerly is called Micronutrient Initiative, is an organization based in Ottawa, providing 75% of the world vitamin A capsule. So they are funded by Global Affairs Canada. So we have looked at the impact of too little vitamin A or vitamin A deficiency. Now we can look at another spectrum of the extreme, which is the overdose or vitamin A toxicity. When vitamin A is taken at an extremely high dose, in the form of retinol especially, has been shown to be toxic. It acts as a teratogen, a compound causing the birth defect. So because it might cause birth defect and it's because of toxicity, the prenatal supplements normally contain relatively low amount of vitamin A, and the majority of them, as we see here, is a form of beta-carotene. And this is because beta-carotene has not been implicated in birth defect. And looking at this formulation of this prenatal supplementation, we also see that folic acid and kept in 1,000 microgram, and which we discussed earlier this month, is low enough to prevent masking the vitamin B12 deficiency. And also to prevent the vitamin B12 deficiency, we can see B12 is also added here as well. Maybe we can take a 5 minutes break and we can go back for our vitamin D. Maybe let's come back at 2:11-ish. So since the role of vitamin A in immune system is pretty important, I noticed I may went too fast, or maybe we can like just like restart this again from this immune system. I'll just spend more time on it. So in addition to controlling the gene expression we saw in previous slides, the vitamin A is also required for the differentiation of the cells in the immune system. So from a precursor, which is a cell, actually the parent of the cell, it require this vitamin A to be able to divide it into like later version offspring cells. We call it lymphocyte. We call it phagocyte and lymphocyte, which are both white blood cell in our body to help against a pathogen in our body. So like we said before, vitamin A play an important role in both innate immune system, immunity, and acquired immunity. In the innate immunity, the white blood cell that's circulating in our body are crucial for the immune response. And under this white cell umbrella, we have phagocyte and lymphocyte. So phagocytes, a good example of the phagocytes is the macrophages. So they engulf, when there's a harmful organism or pathogen in our body or specifically in our blood, this type of phagocell will directly to go engulf this pathogen, and it will try to break down through specific mechanism involving antioxidant system, which we won't talk too much about here. And after the pathogen is broken down, the part of the pathogen, also we call it antigen. So it's a specific protein, like a name tag of this pathogen, will be presented to our next set of cell, lymphocyte. So the this side of the cell defense system, we call it innate immunity. And once the phagocyte present this antigen to the lymphocyte, once it sees the name tag or antigen of this specific pathogen, it will secrete antibody. And what this antibody does is to recognize and bind the antigen presented in the harmful organism. So next time the body sees this specific pathogen, instead of go through this phagocyte process again, it will directly secrete this antibody to attack this pathogen. So that way it's more sufficient and effective. That's why vitamin A is important for all this process, because vitamin A, because those two cells, lymphocyte and phagocyte, they are differentiated from the white blood cell, and vitamin A is actually important for this step. So in conclusion, vitamin A is really important for both innate and acquired immunity. That's part of the vitamin A and the immune system. So next, we have another fat-soluble vitamin, vitamin D, which we also call it sunshine vitamin. So vitamin D is one of the vitamins that's actually we can synthesize in our body. Specifically, you may all heard about cholesterol. And for cholesterol, we have 7-dehydrocholesterol, which is a derivative of the cholesterol. And when our skin is exposed to sunlight, this 7-dehydrocholesterol will be converted to vitamin D3, also called cholecalciferol. So you may ask if we can synthesize vitamin D, why we still call it a vitamin, or why we still need it from our diet. That's because during the winter, especially in latitude greater than about 40 degree north or south, which showed in those gray area in this map, there's not enough direct sunlight for vitamin synthesis to occur because because of the lack of sunlight during the winter. Furthermore, the vitamin D synthesis is impaired when people spend most time indoor, as we tend to do more in modern society. And also, the biosynthesis in the skin of the vitamin D declines with age as well. And another thing is during winter, we tend to cover the majority of our skin by clothing. So that's why most people require dietary or supplementary vitamin D to meet their requirements, especially during winter. The main function of the vitamin D is to maintain a healthy bone. So vitamin D is essential for maintaining calcium level in the blood. It does so in three ways. It by increasing the calcium absorption in the small intestine, which is a main site of the calcium absorbing site. And also it does so by increasing reabsorption of the calcium in the kidney, and so less calcium will be lost in the urine. And also it does so by increasing the breakdown of the bone or reabsorbs of the bone to release calcium. So the last point may seem a bit counterintuitive to improve bone health, but in fact, vitamin D breaks down the bones when calcium is absent in the diet, because maintaining a normal calcium level in the blood is essential. Additionally, vitamin D also stimulates bone formation when calcium intake is adequate. So when you consider all those three functions together, vitamin D really is a promoter for bone health. For this figure on the right, you don't really need to know all the details. Just need to remember the main function of vitamin D on bone health and what's the reason of it. That should be sufficient. And in terms of how vitamin D regulates our genes, it's pretty similar we saw with vitamin A in the earlier slides. It's basically enter the cell and bind to its carrier or receptor, and they drive a regulatory unit to drive gene expression and later affect protein translation. So how vitamin D increase the calcium absorption from small intestine? Well, it does so by promoting the expression of the gene that encode the calcium transport protein in the small intestine, specifically the apical membrane here. This increased amount of calcium from the diet then enter the bloodstream, and in the absence of the calcium transport protein, the less calcium will be absorbed because it is taken up only by passive diffusion. So this transporter is needed for calcium absorption. Vitamin D deficiency lead to loss of bone mineral which we saw before. The rickets, specifically in children, is mainly caused by vitamin D deficiency. And we see example here that where the leg are bowing outwards because the bone is lacking the strength to support the weight of the torso. And ricket in adults is called osteomalacia, but otherwise it's pretty similar, just a different name in different age stages. And another thing to mention that the ricket, especially in children, is likely can be reversed by supplementing vitamin D. Here's a case study at the beginning of the chapter 9, as described in the instance of ricket in the Canadian North. Surprisingly, even in the high-income country like Canada, cases of rickets are still reported every year, although its number is pretty small. In this case, a common scenario for development of rickets is described. A mother exclusively breastfed fed her baby, and which is really the best food for a baby. But the breast milk is low in vitamin D, particularly in the mothers, if the mother's diet is also low in vitamin D, and also especially the mother is in north area, lack of sunlight during winter. So for this reason, the baby developed the ricket, and like I said before, fortunately, this condition can be reversed, can be recovered by vitamin D supplementation to add a diet. And that's why vitamin B supplementation is recommended for infants that are being exclusively fed by breast milk. In recent years, there has been a lot of research suggesting that vitamin D benefits not just for bone, but also it might reduce risk of cancer and maybe other disease as well. The dietary reference intake for vitamin D were first developed in 1997. In order for the recommended requirement for vitamin D to stay current, the scientific literature on vitamin D was reviewed and new dietary reference intake were established in 2010 again. And this demonstrates that how science is being incorporated into existing standard. So the graph here shows the changes that were made as a result of the new review. In 1997, showed in blue bar, and the most recent one showed in like pinkish, red pinkish bar. And in 1997, an adequate intake for vitamin D was established. And you can see the number here is quite low compared to the new standard. The new dietary reference intake or new standard is established a recommended daily allowance, RDA, a concept you might still remember before, and increased significantly amount of requirement for young adults, but also for middle-aged and older adults. So basically across age groups, the vitamin D recommendation is all increased. This new standard was based on, but the standard, the reason increased is all based on the more recent evidence on vitamin D on bone health. And some researchers believe that this number were still too low, and they still recommend even higher, this number to be set higher, because they felt that in order to reduce risk of cancer, and the RDA need to be set higher. There's some evidence over there. But the regulator who reviewed the literature encountered by saying that the evidence for vitamin D on cancer was still weak or still in its infancy and require more research, but the evidence for vitamin D on bone health is quite substantial. So they based on those numbers on bone health. Because the biosynthesis of vitamin D declined with age, Health Canada recommends adults who over 50 to take daily vitamin D supplementation. Another fat-soluble vitamin is vitamin E. So vitamin E actually itself is an antioxidant, just based on its own structure. It mainly resides in the between the double-layer cell membrane. It actually maintains the integrity of the membrane by preventing oxidative stress of the saturated fatty acid contained in the membrane phospholipid. So if you still remember, this is cell membrane, and each structure is a phospholipid, is a phospho group attached to a two lipid chain. So those two chains are mainly unsaturated, so that means they're easily to be oxidized. So vitamin E is mainly reside in between these two layers to prevent the oxidation. it does so by vitamin E does so by neutralizing free radicals by donating one of its own electrons to this free radical to make it more neutralized. And then vitamin E can also receive, after it giving its own electron, it can receive another electron from vitamin. Oops. It can receive another electron from vitamin C. So it's regenerated by the vitamin C. If you still remember at the beginning of the lecture, we mentioned vitamin A and vitamin C and vitamin E. They both antioxidant, and they both work together as an antioxidant system to help protect us from oxidative stress. That's how they work, by donating electrons to each other. Other important antioxidant nutrient is mineral selenium here. It's an important structure. It is important for an enzyme called glutathione peroxidase. It's a structure important for the structure, and the glutathione peroxidase is also another important antioxidant in our body, and it's normally it's also normally neutralize the peroxidase. Of note that if more glutathione or if more selenium in the body or in the diet, the more glutathione will be formed. Thus, the more peroxidase will be neutralized. In that way, we actually don't need that much vitamin E. So, nutrient selenium actually can spare vitamin E or vitamin C in our body. So all those three, glutathione, peroxidase, vitamin E, and vitamin C, they are formed our antioxidant system to protect us from oxidative stress. So that's the main function of the vitamin E. So the antioxidant is an antioxidant. And the last fat-soluble vitamin we will discuss here is vitamin K. Its main source, the main source of it is in the diet, is actually from leafy green, and the chemical form in the food, this compound is called phytoquinine. But in supplement, you may see also in the animal food or in supplement, another form is called manoquinine, also called vitamin K2, and the phytoquinine here is called vitamin K1. The main role of vitamin K is to function in the blood clotting. People who have taken some type of blood thinning medication need to keep vitamin K intake consistent to make sure that the drug dose is correct. There's also been some research interest in the role of vitamin K in bone health, as vitamin K is a coenzyme for an enzyme that is required for synthesis of the protein that regulates bone metabolism. The coenzyme, if you still remember the concept, we introduced that in the vitamin B6 part, so it's important for the enzyme to be able to function. So the required rating for this module is shown here, page 339 and 630 to 631. And what you should be able to do after reading this is to identify its safety and adjusted uses for supplementations. And please maybe spend some time on it after the lecture. This concludes section 2. To assist with your learning, please go through this question and make sure you can answer those after today's lecture. And thank you for listening. I noticed I might went through too fast, but if you have a question, you are welcome to come up. Thank you.
Just a quick thank you to Dr. Yang for coming in today. If you have any questions, I think we can just go ahead.
So it's true selenium, that's what's the important component of
So I guess what we can do with our 1 hour tomorrow is to do a Mentimeter question session. So I have questions from previous weeks and then questions about this week's content, and then we can try and answer them together tomorrow. Please feel free to come up if you have any other questions. Okay?
Selenium interaction
Yeah, of course.
with the vitamins. Would you like go over that?
Yeah, of course, yeah. So, here, selenium is actually this mineral.
Yeah,
It's a mineral. So, you want me to go through only selenium part or everything?
I'm fine with those parts.
Okay.
Just the selenium with interaction.
Okay. So, the peroxidase is like an oxidant.
Yeah.
Actually, it will cause, it's like free radical as well. It will cause oxidation stress. So, glutathione or peroxidase is an enzyme, it's a protein actually, containing a selenium, will neutralize like same way, because the free radical,
Yeah.
it need another electron to neutralize it, right? In the same way, it's just another antioxidant to work together. If you don't have this, or if you don't have enough selenium, you probably will need more vitamin E or C. Yeah, because they all working together. Yeah, if you have more of this, you don't need that much of this.
So basically, like this is like not the requisite, like the required nutrient or something. Like if we have these, it would be sufficient. We just need more of these.
Well, I would say we need all of them, because this part is just like one section of how they work together. They all have different functions.
Other functions.
Like for example, vitamin E, people call it like the first time people find it because when they find in rats, when rats don't have enough vitamin E, they are infertile.
Oh.
So yeah, that's probably another example of vitamin E. It's not only because for the time limit we only cover it
A little bit.
as an antioxidant. If you see like for vitamin E and K we only have one size,
Yeah.
but for others we have like multiple.
Yeah.
So I don't know why, but I guess this one is like basic nutrition, right? I guess that's the only thing you might need to know about vitamin E. But if you like take other courses, for example, another course called vitamin E for life cycle, you might learn more about vitamin E, not just antioxidant.
Makes
So
sense.
it doesn't mean if we have more of this, we don't need this because they all have different functions.
They have different other
yeah,
functions than
like,
antioxidants.
Yeah, for vitamin C, not only the antioxidant, it also important for collagen synthesis, like our skin.
Yeah.
Yeah, so same thing here. Selenium is also has other functions. Yeah, that's an example. Like
Makes sense.
we don't have to. It's not like we have this, we have this.
Yeah.
We need all of those.
We need all of them.
Yeah, just not a. Because
And
it's just
this-
fair shot.